Hi, my name is Sergi Borrego from the Hospital Clinic of Barcelona, and I am glad to have this opportunity to share with you our work. Use of brainstem white matter predict onset of motor neuron symptoms in C9orf72 expansion carriers, a GENFI study. As you already know, the pathogenic C9orf72 expansion is the most common genetic cause of frontotemporal dementia and ALS. We also know from neuropathological studies that C9orf72 carriers show important white matter neurodegeneration, especially in the corticospinal tracts. For that reason, the aim of this study is to analyze if white matter volumes of the brainstem in presymptomatic and symptomatic C9orf72 carriers may be helpful in predicting the onset of symptoms. For this study, we focus on the brainstems because it has a high proportion of white matter and it is easily measurable by cerebral MRI explorations. We studied 229 participants from the GENFI cohort. This cohort includes 41 patients with frontotemporal dementia due to C9orf72 pathogenic expansion, 11 subjects with ALS due to this same mutation, 102 asymptomatic carriers, and 75 controls. All participants underwent a T1 MRI acquisition that allowed us to estimate the white matter proportion in the whole brainstem and age of these regions, the midbrain, the pons, and the medulla oblongata. In the group comparisons, motor neuron disease and FTD C9orf72 carriers showed lower white matter ratios in the midbrain, pons, and in the medulla than asymptomatic carriers and controls. On the other hand, no differences were found in any brainstem region between asymptomatic carriers and controls. In that sense, both cognitive and motor impairment were inversely correlated with the brainstem white matter ratios. When performing linear regressions to assess the relationships between the brainstem white matter and age, we found that both C9orf72 carriers and non-carriers showed a mild loss of white matter ratio over the years, with both groups following similar trajectories until the 5th decade of life, when its trajectories diverge with a greater loss of white matter in the C9orf72 carriers group, suggesting a higher loss of white matter due to neurodegeneration. When comparing by diagnosis, patients with motor neuron disease showed a greater loss of white matter ratio, especially in the medulla oblongata. In summary, we find consistent brainstem white matter loss in C9orf72 symptomatic carriers, with difference related to the clinical phenotype supporting the use of brainstem measures as a neuron aging biomarker. Symptomatic C9orf72 carriers presented a lower ratio of white matter in all the brainstem structures, especially in the midbrain, which can be proposed as a biomarker of conversion. Subjects who develop the motor neuron disease phenotype present lower white matter volumes, particularly in the medulla oblongata, than those who presented with the FTD phenotype. This finding suggests the white matter ratio of the medulla might be a predictor of the motor neuron symptoms in C9orf72 carriers. Thank you for listening.